Okay. Good evening, students and faculties. So today is the third day of our CBSE Spark Minds webinar. Today we have biology class, the complete 4 hours. So we start in a moment. Sir, we'll wait for some more attendees to join and then we'll start.
Meanwhile, we can play our videos, Aniket.
Yes, sir.
Time. The time proves that we have come a long way to get where we are today. Dayanand Sagar University, with the legacy of 6 decades, had contrived to the achievements today and is throwing light on the way forward. Our visionaries understood the need to learn and know, to do and grow, to twiddle and tweak, creating the best of the thinkers, creators, innovators, and inventors. In fact, we believe in achieving more than ever before, instilling the commitment to strive continuously to serve the national and global needs. There are challenges we can't ignore, but remember, we are of humankind, and every objective is achievable by setting our minds, and Dayanand Sagar molds you to achieve the objectives in an environment that enhances creativity and innovation. If we all embrace the spirit that there really is no limit, if we rise to this occasion and come together across nations, if we integrate and ideate, we can make the future great and live the dream. Our legacy of education takes its root from our founding father, late Shri Dayanand Sagar, a visionary, an educationist, a powerhouse of knowledge and a believer in transforming the world, besides a leader par excellence. The institution has contributed to educating the world for over 60 years, carefully building alumni of over 100,000, including accomplished professionals, sports personalities, celebrities spread across the globe. We stand strong as a top-rated university, having 4 campuses with 350 teaching staff, educating 4,400 students with pride-filled and powerful alumni network across the globe. Dedicated faculty members who work across disciplines, collaborating and lifting us as the top university in the world, empowering the rising generation and nurturing entrepreneurs. Dayanand Sagar University. Live the dream.
Okay. Yes, sir. We are good to go live, sir, now. And we are okay to start the session. But, sir, I wish we could wait for 3 more minutes and we'll wait attendees to join.
Okay.
Yes, sir. Uh, Prashant, sir?
Yeah, Nikesh, tell me.
Yes, sir. Sir, we can start now, sir.
Uh,
Yes.
Good evening, students. Let us start today. New topic: strategies for enhancement in food production. Okay. Now, please keep notes and NCERT textbook with you before starting. Okay. Okay. In this chapter, the CBSE pattern, how they are bifurcated means two half they are made. The first half they are explaining about animal husbandry, so rearing of domestic cow and poultry, fisheries,
Excuse me,
honeybee,
sir.
all those. Yeah, tell me.
Sir, your
Hey,
voice was too low, sir.
okay, 1 minute. Okay.
Yes, sir.
Now, is it
Yes, sir.
clear?
Now it's proper. Yes,
Uh,
sir.
Okay. So in this chapter, there are two halves. The first half consists of animal husbandry, in the sense rearing of domestic animals such as cow, poultry, fisheries, honeybee, like this. Okay. The second half is totally into plant breeding techniques. Okay. So where there are two techniques given in detail: conventional breeding and mutational breeding. And based on these two techniques, we are going to study about disease resistance, pesticide resistance, okay, and insecticides resistance. Okay. So let us start this chapter now. Just a minute. Now coming to this introduction, the initiation taken by the breeding techniques, okay, whom we call it as father of green revolution in India, father of green revolution in India. Okay, so he will take this initiative in enhancement of food production in India. So in his technique, it consists of genetics, plant breeding, radiation research establishment techniques, cytogenetics techniques, okay, which was all under the control of IARI. Okay, now let us come to the next. Not only that one, the novel work done by the Swaminathan was rice hybrid variety. It was developed Swaminathan in basmati rice. It is the major achievement, okay, by the Swaminathan. Next. Okay. Now, Orlog was called as father of green revolution for the world. Swaminathan is for India, related to India. Norman Borlaug was called as father of green revolution for the world. Okay. Norman Borlaug means semi wheat he only discovered. Student, this is given in the first page. Before first page, they have given this. Okay. Introduction part they have given. Okay. Next. Next. So let us go to the next slide. Just a minute. So same thing, Padma Bhushana, these are not so much important. Okay. Now coming to this chapter, as I told, animal husbandry and plant breeding technique are the two key factors in this chapter you are going to study. Okay. And here you are going to study one more concept that is called as mutational breeding, also we call it. Okay. What is this mutational breeding? Changing the DNA sequence of the plant. And the second technique is tissue culture technique we are going to study. In tissue culture technique, we are going to, okay, just a minute. Okay. In tissue culture technique, we are growing the plant in the laboratory or in vitro condition. So that's the specialty of tissue culture technique. Recombinant DNA technology, RDT also we can call that. In recombinant DNA technology, we are manipulating the DNA of the organism. But here, all these three techniques are applied to the plants only. So student, they ask for 3 marks. In plant breeding technique, what are the 3 techniques implied? Okay, in plant breeding technique, answer is mutational breeding, tissue culture, and recombinant DNA technology. Now, next topic. Let us take the first one, plant breeding technology. Let us discuss about plant breeding. Okay, in plant breeding technique was first introduced from the ancient period. What is this traditional form of agriculture? Okay, in olden days, what they used to do, they normally used to have wide variety of species, plant species, because 100 or 200 years back, if you back, their technology was not so well developed. So all wild variety plants were cultivated in the field. So this is called as traditional farming technique we call. Okay. Now, here, as the days is passing on, the population is rising. Okay. So the population is rising means definitely the food source is required more in number. So to improve this food source, practices and increase in the yield is also very, very important. So this is the introduction part. So from traditional farming, we have switched to plant breeding technique. That is mainly because of the population. That is the reason. Okay.
Excuse me, sir. So there is a question from Arbia for you.
Now, this, yeah, tell me.
Yeah, sir. So Arbia is asking, sir, this chapter is for NEET. It is related for CBSE board, right?
Yes, Cbse, first page only. Okay, wait. I'll say the page number also. What she's saying is correct. Huh? Let me give this one. See, page number 170. If you come to the NCERT textbook, Arabia, here, page number 170, here, they have given introduction part. Take farming to plant breeding technique. How did they switch? Okay. So that is what given here. Page number 170. Okay. Shall I continue? Doubt clarified?
Yes, sir.
No. No.
Arbia and other
Let us
students, if
go
you
to
have any doubts, you can raise your hands. I can allow you permission to talk. You can directly ask sir also, whatever your doubts are. Thank you. Yes, sir. You can continue, sir.
So we are increasing the yield. That is by plant breeding technique. Okay. Next. Now, when this plant breeding technique started, then only green revolution concept was majorly introduced. Okay. So Swaminathan was the famous scientist. Again, he was the scientist who has improved this green revolution technique. Okay. Now, Norman Borlaug was father of whole world. He initiated this green revolution. So in green revolution, what was the plants which were targeted? Wheat, rice, and maize. These were the species keenly targeted. Okay. These are meant for human consumption. Okay. Next. Now let us come to in plant breeding technique. Okay. There are two methods, as I told before only, conventional breeding and another one is mutational breeding. Now, why do we do this plant breeding technique? One is improving the quantity of the food. Second one is better yield. And third one is disease resistance. So these are the key concepts. Okay. Now, coming to this one, in this slide, In the wild variety, see, if we go 200 years back or 300 years back, wild variety, ancient technology was used. Okay. Now, they have used varieties of human management techniques. Okay. So today, majority of crops are derived from domesticated variety. The meaning of this one is wild variety, they have manipulated the DNA. So after manipulating, hybrid varieties are obtained for the higher yield, high nutrition, and resistance to diseases. Okay. So this was done in both plant as well as animal. Okay. Now, let's go to the next slide. Not only in this plant breeding technique, there were few other technologies were used. Okay, genetic, molecular techniques, and tissue culture were also this plant breeding technique. Okay, so in a simple way, they want to improve the yield and quality, increase in the population, human population, and not only that, they wanted to obtain a hybrid variety of crops which were grown. Okay, which can be resistance for the stress, pathogen, and all insects resistance were also the main factor of this one. Okay. Now, let us start the plant breeding technique. Okay. It was initiated by government institution and some commercial companies were involved in this plant breeding technique. Okay. So this is very important, students. This is a 5 marks question. Okay. 5 marks question. What are the steps involved in plant breeding technique? The first one is collection of variability. Okay. Where you collect different varieties of plants, evaluation and selection of parents. In that variety, 2 parents you must have to select, which is disease resistant. Third one is crop hybridization among the selected parents, in the sense crossing between the two superior quality. And the fourth one is selection and testing of superior recombinant. That means after hybridization of selected parent, you will get a recombinant species, that is progeny we call. Okay. So we must have to check the progeny, select and testing of the superior progeny or recombinant we must have to do. Then once it is passed, testing, releasing and commercialization of new cultivars. Okay. 5 marks question. What are the main steps involved in plant breeding? Okay. Here, one more one by one I will start. Now, one step I'll take. Collection of variability. Okay. So what is the mean I will teach? Okay. Imagine, okay, in India, imagine just a rough example where I teach. You must have to explain also. Simply you cannot write the heading, collection of variability, like that you cannot say. Imagine in India, there are 10 types of mango. Varieties of mangoes are there. I know more than that it is there. So in the 10 varieties of mango, from North India to South India, you bring all of them in Karnataka and you just grow all of them. So this is called as collection of variability. Okay, student, this topic is given in page number 171. Okay, here one important word is there. Germplasm collection, you can see there also in this topic only, collection of variability. What is germplasm collection? Collect, one more question they will ask in theory exam. Collection of plant parts, either leaf, root, or whole plant, or seed, is called as collection of germplasm. Please make a note question. Okay. Now, so as I told, there are 10 varieties of mango. Bring all of them, grow it in Karnataka. That is called as collection of variability. Okay. preservation. Next, see, germplasm collection. These are the seeds collected. Or you can collect whole plants. Is it clear? Next, second one. Evaluation and selection of parents. What is the meaning of this one? Now, evaluation means check all the 10 mango plants for 3 to 4 years. Okay. In that one, once they start yielding the fruit, you select only 2 parents on the criteria of infection, okay, stress resistance. These are the 2 major criteria. So you should select 2 parents, and third one is yield also. High yield and nutritive value of the fruit. So select any 2 mango species which you have grown from the 10. You select only 2, and from those 2 should be resistance, disease resistance, high yield, and nutrition value of the fruit will be more. Okay, so this is the important. Here. In this, they are shown as a rice, Oryza sativa as an example. They are showing. Okay, I am giving example of a man. Okay. Same thing, evaluation and selection of the parents. Now let us come to the third step. Cross hybridization among the selected parents. What is the meaning of cross? You have selected 2. Now cross hybrid. What is cross hybriding? Fusing of the gamete of the 2 plant cells is called as hybridization. Okay. So this is the artificial technique, man-made technique. Okay. So you should do cross hybridization between 2 mango species which you have selected. Transferring of pollen grain to the stigma of the another plant, same species. So this is called a hybrid variety. Okay. Now, what are the major criteria of doing this one? High protein quality or quantity yield and disease resistance. Next. This is the artificial hybridization technique. Is it clear? Transferring of pollen grain to the stigma, artificial hybridization technique. Okay. Where emasculation, bagging, that is in the second chapter, reproduction of foreign plants, there you study. Here also, this technique is included. Okay. After this, once you have done the cross hybridization, what you are going to do? Now it undergoes double fertilization. After fertilization, what you are going to do? Selection and testing superior recombinants you are going to do. Means after fertilization, fruit formation and seed formation. Select the seeds, okay, and try to grow those seeds, hybrid seeds, okay, and test for the disease resistance, okay, yield resistance, okay, and nutritive value of the fruit. So this is called as careful scientific evaluation, we call. Careful scientific evaluation of the progeny, we call. Is it clear? Now. So in this step, the plant that are superior to the both the parents. Okay. Now, let's go to the next slide. Fifth technique. So once the hybrid variety is tested, you are going for testing, release, and commercialization of new cultivars. Meaning of this one, you grow that progeny separately in the field. Okay. Once you have done the testing, the quality wise and disease resistance wise, what you must have to do? You must have to again grow in more in number to obtain more progeny. In the sense, seeds. Once you get those seeds, you release it to the farmers or new cultivars. Okay, so this is what releasing. See, they are doing testing, disease resistance, all those things they have done the testing. Now evaluation technique, because simply they cannot release into the market. Okay. Farm testing they will do, all those things. Okay. Here. Local variety, hybrid, new cultivar, new variety. Okay. Now, coming to this one, this point is given in your textbook. Okay. I will set page number, yeah, 172. Go to page number 172. One point is there. Actually, this has come for the exam also. Okay. Now, coming to this one, what is the GDP, Indian Gross Domestic Product? If you take agriculture, 33% we are getting GDP from agriculture sector in India. Please make a note. Okay. 33%. One mark question also they can ask this question. Page number 172. Okay. Page number 172. Here, one more point, I think 62, this is also is there in the textbook. In India, agriculture employs nearly 62% of the population. So 62% are farmers as per the report given. Okay. So this is there in the NCERT textbook. So this one before independence and after independence also these questions have never raised. Okay. India after independence it was thriving for the food. Okay. Because there was very less transportation. Okay. In India. So now India started, once the technology started implementing in 1960s, they started producing high variety, different varieties of yield, rice. Okay. Where immediately the food production increased and the need of people of India food were also increased in this technique. Okay. So this part, wheat and rice was main focus by Indian government actually. Okay. In green revolution. Now let us come to the some Indian hybrid cross field varieties. Let us go. So these to the page number NCERT page number 173. If you come there, okay, can you see this Sonalika? Sonalika. So this is a hybrid variety of wheat developed by India. And one more is Kalyan Sona. Kalyan Sona. Sonalika and Kalyan Sona was the hybrid variety developed by Indian scientists. Okay. The speciality of this one is semi-dwarf variety. Actually. So these are semi-dwarf variety we developed by the India. Like that, they have developed maize and garden pea also. But maize they have not given. Okay. And garden pea variety in NCERT book, it is not given. Okay. So now this is very important. So wheat production increased from 75 million tons. This sentence is there in your NCERT book. 75 million tons from 35 million tons to 889 million tons. Sorry, I made a mistake here. Wheat production from 11 million tons. Before Green Revolution, only India was growing only 11 million tons. Okay. Now it has increased to 75 million. Like that, rice from 35 million ton has been increased to 89.5 million tons. Okay. Now, this is all because of Green Revolution and plant breeding technique. The speciality of all these are semi-dwarf variety, either rice or wheat if you take. Okay. So, like that, Norman E. Borlaug at International Center of Wheat and Maize Improvement in Mexico developed semi-dwarf variety wheat. The meaning of this one is India did not develop first semi-dwarf variety. It was Norman who developed, okay, in Mexico, wheat and maize, semi-dwarf variety. And India took the technique from them and then they started developing rice and wheat in India, semi-dwarf variety. This is what the meaning of this slide says. Okay. Next. In 1963, again, Kalyan Sona, Sonalika, or high yield and disease resistance was introduced all over the wheat growth belt in India. Again, this one. Let us come to this one. This is very important. 2 marks question for theory. Okay. Here, semi-dwarf rice variety. I was explaining about wheat. Now, which country first developed semi-dwarf variety rice means it is Philippines and Taiwan. Philippines developed IR8. Okay, IR8. The institute was International Rice Research Institute. Okay, located in Philippines. And the same semi-dwarf rice variety was developed by Taiwan also. And they kept the name as Taichung Native 1. So please be careful. IR8 is from Philippines. Taichung Native 1 was developed by Taiwan. Is it clear? Now, same thing, IR8. Can you see this one? In India, if you come means, okay, 1966, this was what is Jaya and Ratna, semi-dwarf variety. India developed Jaya and Ratna. This was developed by India, okay, in 1966. Now here, sugar cane, okay. See, in North India, okay, in North India, they grow sugar cane. In South India also, they will grow sugar cane. The speciality of that one I will explain. In North India, the name of the sugarcane species was Saccharum barberi. Saccharum barberi. Please remember this scientific name. In South India, Saccharum officinarium. Okay. Page number 173 in NCERT textbook. You can open page number 173. This is a 3 marks question, theory question. In India, Saccharum barberi was grown. The speciality of this one was the stem thickness was very small. Hence, the sugar yield was very less. Okay, as the thickness of the stem was very less, hence the sugar was very less in this one, North India sugar cane. Coming to the South India, Saccharum officinarium, the stem thickness was big. Hence, the sugar yield was Indian cross hybrid variety can be grown in North India, and hence the stem thickness increased, and the yield of the sugar also increased in the North India. How is this possible? Plant breeding technique. Is it clear? Plant breeding. Next. So this is South India. Saccharum. Both were both in like that. Millet students in NCERT, they have not given which, so we are not going to discuss in deep. In me, what they have given, they have developed the hybrid variety of maize, jowar, and bajra. Like they have given. This was successfully by hybridization technique. Resistance to water stress. This is important. Okay, what is it here? Developed by India only, but they have not given the variety in the NCERT textbook. But what was the 3 varieties specifically for resistance to water stress? Next one is plant breeding for disease resistance. Okay. Now, what are these diseases? These are the pathogens which cause infection to the plants. What are those type of pathogens? It can be fungal, viral, okay, So these are the different types of pathogens which cause infection. Once the infection is there, the yield of the crop is drastic. Okay. So these are the different types of infections which we are facing. Okay. So heavy crop loss will be there from 20 to 30%. Okay. Now, so in this circumstance, what farmers can do, they cannot do anything. But plant breeding technique scientists, they have to overcome this disease resistance and increase the food production. So how to do this one? Plant breeding technique. Okay. Now, hybridization technique. So what they are doing, this is given actually. They'll take a plant, healthy plant of two same species. Okay. They will hybridize it. Once they hybridize it, they get a new variety. This new variety will be more dominant, will be having more dominant characters. Same like genetics we are studying. So once this dominant character appears in the plant, resistance to the disease also is. Once they in this two types, fungal infection can be bacterial infection.
Sir, there is any network issue in your area, sir? Hello, Prashant, sir.
Yeah, network issue was there. Network issue. Yeah,
Acha,
just a
okay.
minute. Just a minute. There was a network issue from my side.
Okay, sir. It's okay.
Share this. So this is very important. In your NCERT textbook, they are given few fungal diseases. Page number. Actually, where is the page? Few fungal. You can see page number 17. Method of breeding for disease resistance. Okay. The fungal disease they have given. So this is the brown rust. Very important. What are the fungal disease they have asked in the Cbse question? Okay. Brown rust. Sugar rot of sugar. Red rot of sugar cane. Red rot of sugar cane. Next. Late blight of potato. So very important question. That's Cbse board examination. Fungal. These are all fungal infections. Is it clear? Next. Bacterial infections come to the. This is also given. Actually. Black rot of crucifers. This is crucifer. Citrus canker. Can you see? These are the two bacterial diseases observed. Next, viral diseases. Tobacco mosaic virus. You can see the patches, yellow color patch. Turnip mosaic virus. Okay, black patches. Now, let us come to the methods of breeding of disease resistance. conventional method. Okay. What is this conventional method? See, as I explained, this can come for 3 marks also. Same thing, plant breeding technique, 5 steps we have studied. What is that 5 steps we have studied in plant breeding technique? Selection of variability, first step. Evaluation and selection of parent, second step. Third one, cross hybridization among the selected parent. Fourth step, selection and testing of recombinant, superior recombinant. Fifth step, this is what we are studying in plant breeding technique. The same technique you will follow.
Excuse me, sir. Sir, I
Okay,
think your
just
network is
write the
unstable,
heading.
sir.
Okay, this is what conventional breeding technique is. Unstable, huh?
Yes, sir, because your voice
Now,
is breaking in between.
now, yeah,
Uh,
yeah. Let me show
it's
it.
there, sir,
Correct.
still.
Again, it is breaking. Voice.
No, sir, now it's clear, but your video is lagging. Maybe, sir, you could do it with turning off your video.
Okay, turn off the video.
Yes, sir.
Okay. Now,
Yes, sir.
coming to this conventional breeding, same plant breeding technique only you must have to follow. Is it clear? The same 5 steps you must have to write. Now, let us go to the next one. Second one, mutational breeding. Students in NCERT textbook, they are given gamma rays. You must have to use gamma rays. See here, radiation they are showing on the seeds. Okay, that seed they are growing. So this is called as mutational breeding. What is mutation? Sudden change of the genetic material alters phenotype and genotype of an organism is called as mutation, we call it. Okay. So if you do mutational breeding, sometimes it will be good, sometimes it will be harmful also. But for the good sake, we must have to do this one. If it is a harmful, we must have to burn the crop itself. Okay. Now, and this is to get disease resistance from first virus. So okay. Friends, can you hear me now? Yeah. Now coming to this one,
Sir, your screen is not visible, sir. You have to reshare your screen, sir.
we share. Okay.
Yes, sir, it's visible now.
Visible now? Network issue from my side only. It's a
Yes, sir. I get that, sir. It's okay, sir.
Now, students, this is very, very important, especially what is the disease resistance plant produced in wheat. The name of the variety is Himagiri. It is resistance, leaf and stripe rust and hill blunt disease it is resistant. So make a note, name of the crop, variety developed by the India, and which disease it is getting resistance is important. Okay. Brassica is the another crop. Pusa surnim, currant rye also we call that one another name. White rust disease it is resistance. Like that cauliflower, Pusa shubra, Pusa snowball. Now here, one peculiar thing is, what is this Pusa? This is the place in Delhi where Delhi University is located. So they have kept the name of the plant species based on the university name. Okay, like Indian Institute of Science, Yeshwanthpura we call in Bangalore. So like that in Delhi, Pusa is the place where Delhi University is located. Okay, now let us come to the cauliflower, Pusa shubra, Pusa snowball K1. Shubra can overcome and snowball can overcome black rot and curly blight black rot. Cowpea, okay, Pusa common, it can be resistance from bacterial blight, and chili, Pusa sadabahar, chili mosaic virus it can overcome, tobacco mosaic virus, and disease it can overcome. Okay, next. This is very important. Again, repeated. And stripe rust hill blend. Southern currant dry, okay, brassica, which is resistance to white rust. Pusa shubra and snowball cauliflower variety, resistance to black rot and curl blight black rot. Next, Pusa common. This is the which is resistance to bacterial blight. Next, Pusa sadabahar species, resistance to chili mosaic and tobacco mosaic and leaf curl resistance. So this is all possible by conventional method only, all these things. Okay. Next, induced mutation. Gamma rays. I said exposure to the gamma rays causes mutation in the plant species where good variety of species were obtained. Okay. Yeah. See this word, this sentence is very, very important. Gamma radiation is important in just exposure of plant to the gamma radiation, you will get a mutation breeding plants. This is what it is explaining. Okay, exposure of plant to gamma radiation, we get mutated species. Okay, and this technique is called mutation breeding. Okay, now this word is given in your NCERT textbook. In mutational breeding, which plant did they do? Moon bean, but they have not given the variety name. This has come for Cbse question. In mutational breeding, moon bean was resistant to yellow mosaic and powdery mildew were induced by mutation breeding. This only sentence is given. One more question this is for theory. See this, moon bean. Okay, next. Let us come to this one. They have said in the NCERT book, several other varieties were also obtained. Okay. Next. This is also given. One Prabhani Kranti. Compulsory. I think I am expecting finger with all. Okay. Now, what is the scientific name of wait, my internet is student, can you see this slide? Hello?
Yes, sir, we can see, but sir, your voice is breaking, sir. I think there is a lot of network issue in your area, sir.
Achha. Yeah, my area is little bit here. Okay. Now coming to this, I'm expecting this question. Windy, this thing. What is this? Please, this is very important. The scientific name is so this is one question. Another question, yellow mosaic virus. Please make yellow mosaic virus. Okay. So these are the things. So resistance from yellow mosaic virus. The scientific name is Abalmastus esculentus. Okay. Next slide. See, this is Prabanta. You see. Now, so these were all techniques related to disease resistance, okay, where wild species of the plants were exposed for the conventional and mutational breeding technique. Now let us go to the second type, insects and pest resistance. Okay, insects is the major problem. Okay, because plant is the major food source for the arthropodas, especially insecta. Okay, now you can see here infection of insects. Can you see? Oh, now. Now, students, this is a very important question. Sometimes plant have developed defense mechanism against the insect. What is the defense mechanism developed? I'll say the page number. Okay. Okay. This has come in this one, last chapter, last unit, thorns. Okay. I'll say this one. So what are the defense developed by the plant from the host, from the insects? Actually, this comes in the last unit, okay? There, this defense mechanism, one is morphological character, development of thorns, you can see. Second one is biochemical character, producing the chemicals so that the insects are not at all attracted towards hairy leaf, so that insect will feel difficult to consume the leaf part. It just sits in cotton. These are the resistance, the natural resistance developed by the plant. Okay, predators, I think there it comes. Correct? Huh? Am I right, student? This is also one of the natural resistance developed by the plants. Seeds, weed, okay, okay. Page number 175. Only few points have been. Page number 175, NCERT book there. Okay. Now, resistance to gesseits in cotton, resistance to curl leaf beetle in wheat. So, what is this resistance developed? This cotton and wheat have developed a hairy leaf, hence gesseits will not attack cotton plants and cereal leaf beetle will not attack the wheat. You can see here. Third one, solid stem. Okay, if it is a solid stem, so stem sawfly cannot attack this species. See, this is a solid, hard stem. So sawfly cannot bore inside the stem. And smooth leaf, in the sense, cuticle. So hence the insect will not get attracted to the smooth leaf. Nectarless. In the flower, if there is no nectar, definitely no insect will try to come to near to that plant. Okay, because there is no nectar. Next. High asperitic acid content. This is very important. If acid is more in the stem region, definitely the insect will not try to consume those kind of leaf. And low nitrogen and sugar content also will not attract the insects. Okay, especially in the maize, so that it can overcome maize stem borer insect. Okay. Now, some of the insect resistance plant developed by Indian variety, very important for annual examination. brassica, rapeseed, mustard is a variety. Pusa, it can be resistance from aphids. Then flat bean, Pusa sem 2 and 3, which is resistance to aphids, and fruit borer. Okra, bhindi or lady's finger, Pusa savarni, and one more variety, Pusa A4 they have developed, which is resistance to shoot and fruit borer. Resistance to aphids. This is all Pusa Gaurav species. Next, Pusa sem 2 and sem 3, flat bean. Resistance to Jessis, aphids, and fruit borer. Bhindi, lady's finger variety. Okay. Resistance to shoot and fruit borer. Next one. Plant breeding for improved food quality. Now, we're going to increase. What is this technique called as? Biofortification. Okay. Now, students, this is very, very important here. Okay. Here, hidden hunger. One more question. Let me open this. See, what is this hidden hunger? See, your mother will prepare 3 food: breakfast, lunch, and dinner. Right? So one day, your mother and father has gone out of station. They have given you the money. You may go and eat in pizza or any burger shop. So till your throat level you have eaten. Okay. Do you think you have consumed a sufficient nutrition in the absence of your parents? No. You may have eaten pizza or burger, but it leads to the hidden hunger. So lack of nutrition inside the human body is called as hidden hunger. Okay. Why? If we have consumed the mother prepared food, never you get hidden hunger at all. Okay. Why? Because in the home preparation, they use fruits, vegetable, all those things. So nutrition value, what is this nutrition? It can be protein, carbohydrate, lipid, vitamin. All these things you are consuming. Okay. So that's why you will never feel any hidden hunger in the home food. Okay. Now. Because of internet. Okay. Next. So this is what hidden hunger leads to. Deficiency of essential micronutrients. Okay. Now this is one more question. What is biofortification? Increasing the nutritive value of a food is called as biofortification. Okay. So what is that nutritive value? It can be vitamin, it can be mineral, it can be protein, fat, or carbohydrate. So increasing this in the food is called as biofortification. Okay. So one of the students. Next. So what is this biofortification aims? Breeding crop with high level of vitamins, minerals, and higher 3 marks question, students. Page number, wait, I will tell you, biofortification. Page number 176, if you go, what are the objectives of biofortification? They will ask like this. What is biofortification? Easily you will write. What are the objectives of biofortification means? You must have to write this: vitamin, increase in minerals, increase in protein, increase in oil content is the main objective of biofortification. You must have any 3 points. If you write, you will get 3 out of 3. Is it clear? Next. Let us come to this one. Okay. Again, there are 2 things here. Okay. Maize hybrid variety they have produced, but they have not given the name of the plant. Hybrid maize variety was developed in which two amino acid contents were more in number or twice in number. That is lysine and tryptophan. Students, 2 marks question. Name the hybrid maize variety which contain twice the amount of amino acid. Okay. What is the two amino acid they will ask? Lysine and tryptophan. Please, it is there in your NCERT book. Okay. Next, second one. Atlas 66, 1 mark question. This is a wheat variety which contain high protein. Atlas 66 is a wheat with a high protein content. Okay. Next. This third point, this is also important. In rice, Rhiza sativa, 5 times increase in the iron content was done. Okay, but they have not given the plant name, the species name. Okay, only they have said in rice, 5 times of which metal was increased. Iron is the answer. Okay, next. Golden rice. It contains vitamin A. This is also beta carotene is important. Please make a note. Golden rice contain vitamin A, you know this one. Beta carotene is next one. Next. This is very important, students. Many vegetable crops released by India, that is in New Delhi, is IARI, Indian Agriculture Research Institute, IARI. They have developed vitamin A enriched crop, that is carrot. Very important. It's there in your NCERT book. Vitamin A enriched, which plant it is? Carrot is the answer. Like that. Vitamin A enriched spinach also they have produced. Vitamin A enriched pumpkin also. Next. Vitamin C enriched crop. Bitter gourd. Next. Vitamin C enriched. Vitamin C enriched. Another one, mustard also they have produced, vitamin C enriched. Tomato, vitamin C enriched. Now, iron and calcium enriched crops together, iron and calcium, potato and spinach. Protein enriched bean crop, broad bean, lab, French bean and garden pea. These were the species. This is given in your NCERT book. They have asked these things. Protein enriched beans crop. Which one is it? Okay. Single cell protein. Now coming to the single cell protein, an organism, there are some organisms present on this earth. Okay, spirulina you can say. These are organisms, these are algae species which containing a protein inside its body is called as single cell protein, SCP. See, a question only answer is there. Single means it is the single cell organism. Okay, protein inside its body. Okay, now why are we studying this one? Because this can be act as a food source. They are the food source for the human beings. Okay, now. Now, another one. The shift from grain to meat diet also creates more demand for cereals as it takes. So 3 to 10 kgs of grain to produce 1 kg of meat by animal farming. Now, percentage of human population suffering from hidden hunger and malnutrition is more than 25%. This question has come. What is the human population suffering from hidden malnutrition? They will ask. Okay, okay. Anikesh, make you host. Yeah? Okay. Anikesh, can you hear me?
Yes, sir. Yes, sir.
Okay. All right. Now, let's go to the next one. See, in case if there is a shortage of meat, cereals, or vegetables, what is the alternate source? The alternate source is single cell protein. Okay. So that is what it says. Okay. Now, best example, spirulina maxima. Make a note of this one, students. It comes under blue-green algae species. It's a blue-green algae species. You can consume this algal spirulina directly. Okay. In a simple way, just a minute. See here. You bring 10 kgs of protein containing vegetable. You bring 10 kgs of vegetable. Keep it on a table. You give me one, you just go to the medical store, ask for spirulina tablet, Dabur company. Consuming this 10 kg of protein is equal to this single tablet. That's the specialty of the spirulina. Okay, it's an algal species which contains full of protein inside its body. There is no carbohydrates and fat at all. Only protein-filled body it is. So that is the alternative source. So these astronauts, if they go to the space, they will never carry fruits, vegetables. Limited amount of fruits they will carry, apple and this one. But they will carry the tablets, spirulina tablets. That will be the food for them in the space. Is it clear? Now, let us go. This is how the species looks. Now, it can be algal species, fungal species, and bacterial species. But in textbook, majority algal species only they are given. How can you grow this spirulina? You can grow it in containing starch, straw, molasses. What is this molasses? In sugar factory, you will get a black color syrup that is called as molasses. Okay, and animal manure, and even in sewage also you can grow the spirulina, single cell protein. Yeah. Now this, so in a simple way, you can grow this algae in a waste materials also. This is how it looks, single cell protein. Okay. Now, other single cell protein, okay, they have given, take yeast, Candida utilis. It is not given in the textbook, so I will not discuss this one. In yeast, another is Saccharomyces cerevisiae, because it is not given, only spirulina is given. So I will not discuss this one. Okay. Now, student, this one is discussed in your NCERT textbook. Methylophilus methylotrophus. This is given in your textbook. This is a bacterial species, protein species. So in textbook, they have given algae species, that is spirulina, and protein species they have given Methylophilus methylotrophus. Okay. So it has been calculated that 250 kg cow produces, means the weight of the cow, produces 200 grams of proteins per day to feed with Methylophilus methylotrophus. If you feed them with this one, so they will produce 200 grams of protein per day. How it will produce from the milk, you will get that source, protein source. So this is what it will do. 25 tons of protein. Is it clear? See, this is Methylophilus methylotrophus growing culture actually. By fermentation technique, they are grown. Okay. And one more, mushroom is also a single cell protein. This is an algal species, fungal species, sorry, mushroom called the fungus species. So this is also single cell protein. Now let us go to the tissue culture. Students, in tissue culture, again, traditional breeding technique was the old technique. Now, because of the demand of tissue culture technique was the new technique developed. Okay. Can you see this one in test tube? We are growing small plants. So they will ask in exam, what is tissue culture? One more question. Growing of a plant in a laboratory condition is called as tissue culture. Do you understand? Be careful. In tissue culture, two types are there, but is not mentioned in the textbook. Plant tissue culture and animal tissue culture. So directly they are discussing about plant tissue culture here. Don't discuss about animal tissue culture at all here. So you should have definition for plant tissue culture. Growing of a plant in a sterile condition in a laboratory. You can add that one also. Is called as tissue culture. One more question. See how we are in a laboratory condition. There is no sterile in the sense, no contamination at all. There is no infection. Is it clear? Now, this is called as tissue culture. Now, this is one more question. Plant. See, you want to grow a banana plant in the lab. Banana plant is there in the field. Imagine you are having a banana plantation in the field. Cut small piece of stem of that banana plant. Bring it to the lab. Once this enters the lab, this banana plant stem enters into the lab, I will never call it as banana plant. I will call it as banana eggplant. So one more question, what is an eggplant? It is a plant used in growing in tissue culture. That is the definition. Once that plant comes into the lab, we call it as eggplant. Okay. The plant used in the tissue culture is called as eggplant or part of a plant. Grown in a lab is also called as eggplant. Do you understand? Is it here? Next, totipotency. One more question. Okay. See, if you cut the plant, the meristematic region or tip of the plant, if you cut, do you think the plant will die? No. Again, it will regrow. So that is called as totipotency. It is the ability of a plant to regenerate from the cut part is called as totipotency, or ability of a living cell of a plant to give rise a new plant is called as totipotency. Is it clear? One more question this is. Next. Coming to this one, nutrient medium. It is there in your book, but very rarely this question has come. What is this? Can you see this conical flask? This is a vibrator machine. In this conical flask, there is a nutrient medium. What is this nutrient medium? Simple way I'll explain. You bring 1 kg of red soil. You bring 1 kg of the red soil. You check what all minerals present inside that 1 kg of soil. Okay. You give me a list. Sir, sodium 0.1 gram present, potassium, like that. There are so many minerals inside the soil. But nutrient medium means there was a scientist called Murasig and Skooks. He developed a nutrient medium which is equal to the soil. So what does it contains? Sucrose, inorganic salts, vitamins, amino acid, growth hormone. Okay, all these things are present, developed by Murasig and Skooks. So in a simple way, nutrient medium is the medium for growing of eggplant, which contains sucrose, inorganic salts, vitamins, amino acids, growth hormone. This is the definition. Okay. Now, these are the chemicals. Inorganic salts. We use 20 salts are there. We mix all these 20 salts, sometimes 40 salts based on the plant. Like soil only, we prepare this nutrient medium in the lab. Next. See here. They not only attract the plant to grow, they will also attract bacteria also to grow. That's why they spoil. That's why the tissue culture is very, very sensitive technique. You must have to grow plant in the lab using nutrient media. By chance, if you are growing plant and microorganism, then that plant will be in a problem. Okay. Now, let us go to the next. So that's why how to kill the microorganism in that nutrient medium. Autoclave is the machine. Sterilization method we do. This we keep. This will sterilize the medium. After sterilization, we will grow the plant inside that medium. Autoclave. These are temperature all neat level. Okay. Next, inoculation. What is inoculation? Placing the explant. Stability. Okay. Now, what is this inoculation? You just put the plant inside the nutrient medium. The technique is called and into the nutrient. Can you see? We are inoculating the plant inside the. Next. Student, this word is there. Callus. Simple answer I will say. How did I get this one? I will say first. Okay. Look here. Take a leaf, any plant leaf. Keep it inside that nutrient medium. Okay. Immediately that leaf will develop into callus. Like this, you can see the photograph. What is callus? Undifferentiated mass of cells. That is the definition. Okay. Now, sir, what is the use of this callus? Take one small piece. These are millions of cells will be there. Just take out one or two pieces from this one. Transfer it to another medium. Each of them will develop into a plant. So in this test tube, one test tube, I can, in case if there is 1,000 callus, I can get 1,000 plants. That is why we call it as micropropagation technique, which I'll teach it. So undifferentiated mass of cells. See, that is what they have. They may develop into shoot and root from the callus. Means they are good treatment of auxins and cytokines based on the treatment of auxins and cytokines. This is important. Organogenesis. Development of a shoot or root from the callus is called as organogenesis. See here. This is a callus. Now it is developed into an organ, leaf. That leaf will further develop into a plant. This is called as organogenesis. Next. Somatic embryogenesis. Students, this is there in your NCERT book. Somatic embryogenesis. Now, coming to this one, what is this? See, I will use system, this is the root system. There are two types of cells. One is reproductive cells. Another one is somatic cell. So these are the two types of cells present in plant: somatic cells and reproductive. Now, what is this reproductive cells means? In a plant, if you are having a flower, that is the reproductive part of a plant. Inside that, you will find reproductive cells. So, flower is the best example of the reproductive cell. Now, come to the somatic cell. In somatic cells, either it can be root system or shoot system except flower. So, it can be leaf, stem, root. So somatic parts means body part of a plant is called as somatic part. Okay. Now let me come back to the topic. Okay. Now, what is this somatic culture means? Growing of a plant, the embryo structure now. So if you get embryo from leaf or root, that embryo we call it a somatic embryo. Means body part embryo we call somatic embryo. Next. Students, this is very micro proportion. Okay. Micro is a small part of a plant taken and grown in large number is called a micro. Okay. Now let us start. Okay. So embryo developed from the somatic part is called as somatic embryo. Now let us go to this. Micro propagation. See, imagine this is the rose plant stem. This is the rose plant stem. When I do tissue culture into 50 pieces inside the tissue culture. So what is micropropagation means? Cultivation of more explants from a single parental plant is called as micropropagation. Here. Next. This is again one more question. Soma clones. Soma clones means somatic cells, you know, body cell is called as somatic cells. So fusing those somatic cells is called as soma clones. So genetically, identical to the original or source plant is called as soma clones. Next one. Meristematic culture. You know, in plant, there are meristematic region, and permanent tissue, complex permanent tissue. Let us forget about that one. Meristematic tissue, shoot tip and root tip. In meristematic tissue, there are three regions: apical meristem, lateral meristem, intercalary meristem. Have you studied in the 11th Cbse? Now, if you grow a plant from the meristematic cells, it is called as meristem culture. What is the use of that one? Virus-free plant you will get from this one. Okay, virus-free plant you will get from this meristematic culture. Is it clear? What is the example of meristematic culture? Example is banana, sugarcane, potato are the best example given in the textbook. Is it clear? So meristematic culture example, banana, sugarcane, potato. The NCERT book it is given. Okay. Now, somoclonal variations. Okay. What is the meaning of this somoclonal variations? Tomato and potato. Protoplast you take, fuse it. Then you will get a tomato. So this is called as somoclonal variations. I'll just show it in a. See again, virus-free culture, meristematic tissue they are showing. So never you will get any viral infection in that one. This is very important, student. What is protoplast? See a photograph. First of all, I will use how to see simple. See, this is it is made up of cell wall and cell membrane, cell organelles inside this one. Now this is called as plant cell. What is protoplast? One more question textbook they have given. This is called as protoplast. Means a plant cell without cell wall is called as protoplast. One more question. Plant cell without cell wall is called as protoplast. Let us go back to the slide. So, cell without plant cell wall. Now, here, protoplast culture or somatic hybridization. I'll come back to the point. What is this somatic hybridization? Fusing of two protoplasts is called as somatic hybridization. Sir, why it is called somatic? Because we have taken somatic cell and somatic cells, body cells, and we have fused it. So this is called as somatic hybridization. Next. So you can see tomato and potato to obtain format. Is it clear? Next. I'll come back to the animal husbandry. Okay, students, I'll use this one, new slide. Okay. Now coming to the second half is totally over. First half. In first half, we are going to study animal husbandry. What is this animal husbandry? It is the rearing of domestic animals. Animal husbandry. It is rearing of domestic animal. One more question. What is this domestic animal? Is it tiger, lion? Like that, domestic animal? Don't write all those things. Cow, sheep, goat. These are the domestic animals. So there is a, be careful while writing. One more question. Okay. Next. Here, if you take whole world, one more question. If you take whole world, livestock means domestic animals. Okay. 70% 70%. Theory question, MCQ, 1 mark. 70% is owned by India and China. India and China. If you want that information, page number 165. Page number 165. India and China is holding 70% of the world, if you see. Next one. If you come to the next one, dairy farm management. Student, this is a 5 marks question. What happened to this thing? Coming to the dairy farm management, 5 marks question. What are the steps involved in dairy farm management? First one is selection of breed, superior breed. First step. How you are going to select? Disease resistance, high yield. These are the superior breeds required. The second one is proper shelter and food. You should have proper shelter and food for this dairy farm management, and expert handler, and you should be aware of this dairy farm management. Next one, regular visit of veterinary doctor. Veterinary doctor should visit, and you should keep the records of all the dairy cattle animals. And next step, proper extraction, hygienic extraction, hygienic extraction of milk. Hygienic extraction of milk so that no contamination. And transport, hygienic transport. You should transport in a hygienic condition. Don't send in a contaminated condition milk. So these are the things given in the book. So these points you should write and explain also to obtain 5 marks. Let us go to the next one. Poultry farm management. In farm management, it is a 3 marks question. How you are going to maintain? Again, a superior breed or superior breed of poultry you should select. Second one is proper shelter and food, same point you can write. Next, expert. Then you should obtain proper products. Hygienic condition, you should obtain the products. Egg or meat you can write. Is it clear? Next one. Let us go to the next point. Students, can you open page number 167 NCERT textbook? In that one, there is a diagram question. In Cbse board, they have asked, name the improved breed of cattle. Name the improved breed of cattle. Answer is Jersey. This will come for this examination. I'm expecting this one. Improved breed of cattle, Jersey. Or improved breed of poultry. The answer is Leghorn. Answer is Leghorn. You can see the diagram. Okay, Jersey and Leghorn. Is it clear? Next, one more question. There is one hidden question inside the NCERT textbook. Okay, name the infection observed in poultry. What is the infection observed in the poultry? Answer is bird flu virus. You must have to write. Answer is bird flu virus. Bird flu virus is the correct answer. Okay. Next up, let us come to the next one. Animal breeding technique. Okay. In animal breeding, there are 2 types. This is 1, 2, 3, 4, and 5 also. See, in animal breeding, there are 2 techniques. Okay. The first technique is inbreeding and and outbreeding. Okay. 2 marks question. What are the two you write? But definition you must have to write. Inbreeding means cross between male and female of the same species with a common ancestor of 5 to 8 generation is called inbreeding. Outbreeding means crossing of male and female of same species with a different ancestor is called as outbreeding. Is it clear? Students, here they can ask in inbreeding, what is the advantage and disadvantage? Disadvantage, pure line. You will get a pure line species. Disadvantage means inbreeding depression. Inbreeding depression is observed. Is it clear? So what is this pure line? Can be also called as homozygosity. Homozygosity means crossing between own brother and sister. What will happen? Same genes will enter into the progeny. That is called as homozygosity or pure line we call. Okay. So here some of the recessive genes are eliminated in this method. Okay. Next one. Disadvantage. Inbreeding depression. In inbreeding depression, the offspring weaker and weaker. Okay. How to overcome this inbreeding depression? Outbreeding you must have to do. Okay. Cross the different ancestral progeny. About outbreeding, let me arrange you answer. Okay, there is all these things. So inbreeding is over. The outbreeding technique. Now outbreeding technique, there are 3 types. Outcross. All right. First one is outcross. Correct. Okay. Page number 1. And cross breeding, second technique. Cross breeding. Third one is inter-specific hybridization. These are the 3 techniques observed in outbreeding. Okay, 3 marks question separately they can ask. Now, what is this outcross? It is a cross between superior male and superior different gender or different ancestors. You are writing. Is it clear? Outbreeding and outcrossing, same definition. Now, crossbreeding, students, here one important thing is that before going to the definition, it is Hisar-Dale. You must have this example. Hisar-Dale. 1 mark, 2 mark question. This one is 2 marks. Now, I'll say the definition. Crossing of superior male and superior female of a different ancestors of different species. Now, merino ram, nightmare eve. Okay, merino ram. That's a different species. Okay, so crossing these two, we get Isaldale. That is called as crossbreeding. Is it clear? Next, last one. Example, mule they have given. Okay. Female horse, male monkey. Any male or crossing between male and female, different genus. Sorry, students, there was a network issue again.
Excuse me,
Hello,
sir.
can
Sir,
you hear me?
yes, sir, I can hear you. Hello. Sir, your network issues there. Sir, we'll give them 15 minutes break and then we'll again start by 6:05.
5? Okay.
Yes,
Okay,
sir.
we'll start at 6:05. Okay.
Yeah, sir. So
Right.
we'll give them 15 minutes break. By the time they'll have their snacks and all. Okay, sir.
Okay.
Students, you can take
I'll join.
15-minute
Okay.
break and then you can join back by 6:05.
Right, Anikesh.
Yes, sir.
Okay. Okay.
So, students, any doubt, any questions related to the lectures or any other related to the sessions, you can ask me.
What do you think a good university should have to give you lifelong learning experiences?
That's a good question.
A good university should have
worldwide recognition program,
innovative pedagogy and interdisciplinary teaching approach,
best-in-class faculties with industry exposure,
modern state-of-the-art infrastructure, advanced lab facilities,
and wide industrial exposure for outstanding placements.
Dayanand Sagar University, a beacon of inspiration, quality, and wisdom. With a legacy spanning over 6 decades, the university is known as the Pride of South Bangalore. Consistently rated as one of the top 3 universities in Karnataka, Dayanand Sagar University inspires students with unique perspectives and knowledge. And now, to inspire thousands more, the university expands its in-demand programs to the Kudlu Gate City Innovation Campus. Equipped with world-class infrastructure, industry-driven innovation laboratories, on-campus medical facilities, and multifaceted faculties, creating a whole new world of possibilities for the brilliant minds in the heart of Bangalore city.
The best thing about this campus is that it is easily accessible. It only takes 45 minutes for me to reach here from Indranagar. Some of my friends come here from Hosur and Whitefield.
DSU City Innovation Campus is so well placed that everyone in the city would love to access it.
The City Innovation Campus now offers coveted programs in commerce, management, arts, design, humanities, computing applications, law, and basic and applied sciences to help students explore the unexplored and discover new possibilities.
This part of the city has always been missing a prestigious campus with these courses, but now with DSU this will be solved.
DSU is the synonym of trust. The university employs industry experts and easily attracts national and international collaborations.
Due to its proximity to the IT hub of Bangalore, DSU is a hotbed for placements and industrial exposure, and also the preferred places for companies to set up the incubation centers.
Experiential learning or practical learning is right at the forefront of things when it comes to student activities.
The relaxed and cohesive environment attained through an engaging, innovative, and industry-aligned curriculum, a technology-driven campus, and innovation laboratories ingrain confidence in students to grab exceptional career opportunities, exemplifying the university's position as a premium university in placements.
In summary, Dayanand Sagar University offers facilities for the overall development, and I request the students to come and explore the opportunities.
DSU persuades us to aim big and to live dreams, and dreams are the most powerful energy. The values instilled at DSU will stay with us forever, and this is how I discovered my career should be.
Come, be a part of this journey to etch the bright future of the next generation. Dayanand Sagar University, live the dream.
Okay, sir. So we'll start now.
Okay.
Yes. So your voice is too low, sir.
1 minute, 1 minute.
Okay.
Ah.
So students, we are starting with the next session.
Aniket,
Yes, sir.
for sharing of screen.
Just a minute, sir.
Ah.
Yes, sir. You can start now, sir.
Right, huh? Okay. Good evening, students. A small topic of strategies for enhancement of food production. Okay. Now, coming to this one. So I have spoken about animal breeding techniques also. Okay. So mule I have explained. Now let us come to the next one. Okay. Control breeding techniques. So this is again 2 marks question. Okay. Control breeding techniques. Animal breeding is a separate explanation. Control breeding technique is another explanation. Okay. In this one, they have given 2 steps. Okay. One is MOET. Okay. Another one is insemination technique. Artificial insemination, correct. 2 marks question. Artificial insemination. Okay. In MOET, there are 2 words. This can be asked for 2 marks also separately. So this is called as control breeding technique. Please make a note. Okay. Animal breeding is a separate method. That's a natural traditional method technique. Now this is the scientific method of breeding. In this MOET, the full form of MOET is multiple ovulation and embryo transfer. Okay. So multiple ovulation can be also called as, this one they will ask, super ovulation. Super ovulation technique also we call. This word is there in the NCERT book. Don't get confused. How to explain this super ovulation or MOET? Simple. Select a cow, okay? Inject follicle stimulating hormone, FSH, follicle stimulating hormone into the cow. So once you inject this one, 6 to 8 eggs will be obtained. 6 to 8 eggs in that cow will be obtained. So it triggers the formation of secondary oocyte in the cow, female cow. Okay. Now this is called as multiple ovulation. Now after this, there is a separate explanation. Now what you are going to do? You are going to select this egg and transfer this 6 to 8 egg in different cow, so that you can place one egg in each cow. Okay. So the transferring into other cows is called as surrogacy or surrogate cow. This they will ask in Cbse. Surrogate cow means it is the cow which receives the egg from other cow. That is called as surrogate cows we call. Okay. Now you are transferring this egg into surrogate cows. Okay. Now after this, fertilization technique is given. Either you can do artificial insemination or mate with the bull, so that the fertilization takes place of all 6 to 8 eggs. So I'll explain once again. Follicle stimulating hormone is injected into the cow to obtain 6 to 8 eggs. This cow is artificially inseminated with the sperm for fertilization or crossed with the superior bull. Stop up to here. This completes multiple ovulation. Now what you are going to do, once the egg is fertilized inside the cow, embryo transfer you must have to do. In embryo transfer, you should be specific. When you get the fertilized egg, 8 to 32 cell stage egg is removed, fertilized egg, and transferred into the surrogate cow. Okay, this is called as embryo transfer technique we call. Okay, and second one, artificial insemination. It is nothing but human made technique where then superior bull sperm is injected into the cow. Okay, superior bull sperm is injected into the cow with the help of the human. Okay, is called as artificial insemination. So this also completes control breeding techniques. Now one more thing, beekeeping. Students, beekeeping. Here you should remember one most important 1 mark question. Apis indica. AP, okay, on mute. Apis indica. INDCA. Now remember this one. Apis indica means Indian honeybee variety. Apis is India. It represents Greek, it's a Latin word. Apis means India. Okay. Word they are using indica. So Indian species. So this is Indian honeybee. And second one, beekeeping can be also called as apiculture. It is also called as apiculture. Rearing of honeybee is called as apiculture or beekeeping also we can call it as. Just a minute. Okay. Is it clear? So 1 mark, what is the Indian honeybee variety called as? Apis indica, and beekeeping is also called as apiculture. Next question, 5 marks question, rearing of honeybee. Okay, it may come for 3, actually 3 or 4 marks actually. Here, a good species of swarm bees is selected, okay, and a proper hive is designed, okay. Third one, green area or flowering area is selected for the placing of the bee. Next, condition is made to obtain before that one seasons. You should proper information of the seasons. People in some so proper information. Every species has particular action of the two products obtained and bee wax. These are obtained from the bee culture. Okay, and points you should write. Okay, it's a 3 to 4 marks question. Okay, next let us go to the next one, fisheries. Okay, students, here little bit information has to be clear in fisheries. Before the fisheries, you should remember actor. This is different, and pisciculture. This is different. One more is there, blue revolution also we can call. Blue revolution also we can call. Now here, first of all, let us discuss aquaculture. Aquaculture is nothing but rearing of aquatic animals. You can take prawn, crab, fish, oyster, anything you can take. That is called as aquaculture. Next up, pisciculture means fish culture, another name, pisciculture. Okay, and next up, in pisciculture, there are 2 types, 2 marks question. Freshwater, marine water. Freshwater, marine water. Here, freshwater, you know, in the lakes and pond, marine means ocean. So those definition is not important. Only types they are given in the NCERT book. And last one, what are the examples of freshwater? This is important. Rohu, Katla, Rohu, and Katla. Katla also we can call. In marine water, if you say take means sardine, hilsa, all these are salmon, pomfrets. These are the best examples of marine fisheries. Okay, very, very important. 2 marks question. Give an example of freshwater and marine water fishes. So you should write example. Don't write all the fish name. In the NCERT book, they have given specified. For example, I will say again, freshwater, cutla, page number 160, carp they have given. Don't write any other things. Don't write catfish, all those things. Only these names has to be specified. In marine water, hilsa, sardine, mackerel, pomfret. 4 of these 4 names. Okay. So this completes about fisheries. So this topic is completed. Let us solve some questions. Okay. Now. Okay. So let me take into this one. Already I have given the answer here. Okay. Sure. Okay. Now let us start few questions. The founder and father of Green Revolution is. So worldwide you should consider. India he has not mentioned. If it is India means Swaminathan, you would have written the answer. Okay. Next one. Which of the following techniques play a pivotal role in further enhancement of food production? All of these. The entire collection of plant or a seed having all diverse alleles for all genes in a given crop is called as germplasm collection, we call. PN1 is the variety of rice, Taichung actually it is. Okay. Rice introduced by Taiwan, Taichung native 1. Okay. Jaya Ratna, wheat, rice variety, sorry. Kalyan Sona, Sonalika is wheat. Jaya and Ratna is rice. Saccharum barberry is the variety of what? Officinarium and barberry, especially in North India sugarcane. Which of the following is a fungal disease? All of this. Next. Tobacco mosaic disease is caused by, the word only says mosaic disease, viruses actually. First you see, you have drawn the diagram also, tobacco mosaic virus. In mung bean, resistance to yellow mosaic and powdery mildew was induced by mutational breeding. Prabhani Kranti. Abalmus esculentus is the scientific name. Immediately, Bhindi or lady's finger. Atlas 66 is a variety of wheat. Next up, beta carotene containing rice variety named as golden rice. Bacterial single cell protein is bacteria. If they say Methylophilus is the answer. Okay, some long answers let us discuss. Here only directly answer. Scientists have succeeded in recovering a healthy sugarcane plant from a diseased one. Healthy sugar plant, how to obtain in tissue culture? It is apical meristem culture where we get virus-free plant. So that is what given here. Okay, how to get a virus-free plant means it is apiculture or meristematic culture. Apical meristematic culture. Describe the procedure the scientist followed by recovering the healthy part of a plant. Simply, they take the meristematic cell, grow it in a agar medium, okay, to get a small plants. Okay. Next one. Name the technology used for crop improvement. See, You can use a lot of technology, but here I have specified micropropagation or tissue culture, you have said. Okay. You can give other technologies also, that has mutational breeding, okay, conventional breeding, okay, and micropropagation technology is all. Out of these things you can give any of one. Okay, I have mentioned micropropagation technology by tissue culture one, I have explained. Okay, here also with answers I have given. Write the scientific name of most common species of honeybee reared. In India, it is Apis indica, as I told before. Mention the kind of area that are suitable for beekeeping place. So which place you are going to keep? You are not going to keep in a concrete jungle. You require a vegetation, okay? Flora, like majority of the flora. It can be shrub, fruit area, or cultivated crops area. Like that area, you prefer to have this apiculture. Mention any two uses of bee wax. This is given in the textbook. Cosmetics and in polish, shoe polish and other polish materials, they use this bee wax. Okay. Thank you, students. So strategies for enhancement of food production is over. Now let me take a new topic. Okay. Just give me 1 minute time. Okay. So principles of inheritance and variation we are going to discuss. Okay. So in unit 2, 12 Cbse. Okay. If you see principles of inheritance and variation, molecular basis of inheritance, and evolution. 3 chapters are there in the second unit. So this is the first topic in the second unit I am going to discuss. Okay. It's a bit lengthy chapter. Okay. Let us start principles of inheritance and variation. Okay. Now, just a minute. Coming to genetics. It is a branch of biology that deals with the genes. Okay. Genes in the sense, what is this genes play the DNA? Okay. They help in studying of inheritance and variation. Okay. In another way, we can call it as heredity also. Okay. Transfer of parental character from transfer of genetic material from parental character to the young ones is called as heredity. Another name of heredity is inheritance. Inheritance, heredity, two different words. Definition is same. Transfer of parental character to the young ones is the definition. Okay. In genetics, we are going to study about inheritance and variation. What is this variation? One more question. Nothing but the parents are different with respect to morphological and genetic character to the offspring. This difference is called as variations we call. Is it clear? Next. See here. Heredity. Transfer of parental character to the young ones. See here. Inheritance. You can see. Variation of parents to offsprings. Have you ever wondered why elephant give birth to only baby elephant? See, this is one topic I have taken. Rare photograph showing DNA relation. Grand old father and right grand old son. See here. More than 5 generations. Almost they are resembling each other. So that is nothing but inheritance. Because of the inheritance. Their features. Parents and offsprings similarities are there, some features, morphology. So this is single gene trait in human. These are all the dominant characters observed in the human. So Mendel experiment, dominant and recessive. These are in the human. They are studied in the NCERT book. They are not given. So I would not like to take this into this one. Okay. So let us go to the directly Mendel experiments. Let us start. So these twins, identical twins, is not important. Okay. Now let us start from here. So genetics deals with inheritance and variations. These are the two important things with genetic deals. Okay. Variation means difference. Okay. So parents are totally different in appearance with respect to the progeny. This is called as variations. Okay. Now, let us know about. Can you see this Indian breed? Sahiwal cow in Punjab. This is genius. Okay. Now, let us go to the Mendel experiment. Okay. Let us see here. Gregor Johann Mendel is known as father of genetics. He was born in Austria. Okay. So one more question. Second question. In which plant did Gregor Johann Mendel work? Pisum sativum. Compulsory question for 1 mark, or sweet pea plant also we can call. He discovered the basic principles of hereditary through pea plant experiments. He published in a book experiments on plant hybrids. Okay. Okay. Now, Mendel worked for how many years on pea plant? This has been there in your NCERT book. 7 years he has worked. That is important. 7 years he has worked on the pea plant. What was Mendel known for? He was known for applied statistical analysis and mathematical logics. Now, in pea plant, what was the contrasting character? How many contrasting characters did Mendel observe? This is very important. 7 contrasting characters. In a simple way, 7 years he worked and 7 contrasting characters he observed in the pea plant. Okay. So what are those different characters observed? Opposite traits we call. Tall and dwarf, dominant and recessive. Seed color, yellow was dominant, green was recessive. I'll come to that in detail. See here, this is a tall pea plant. This is a dwarf pea plant. Okay. Now, this is, can you see this pea plant, tall pea plant and dwarf pea plant? So this is what the Mendel worked. Okay. Now, let us go to the next slide. Okay. See here, in pea plant, the seed were, okay, the seed were yellow in color, which was dominant, and green seed, recessive in color. So garden pea was the material used for his experiment. Why did he select the pea plant? That is the important question. Okay. Why did Mendel select pea plant? Because easy to grow. Hello. Hello.
Hello. Sir, your voice is too low, sir.
Yeah, network issue again. Network issue was there. Okay.
Sir,
Right.
no problem, sir. और कुत्तिया
Okay. So
Sir, the voice is
that
too low,
was
sir.
that's why Mendel selected
Sir, your
three
voice is
plants for
breaking,
his
sir.
experiment. Is it clear? Next, let us see 7 contrasting voice network again network issue. Okay, okay. Now coming to the 7 contrasting character observed by Mendel. Okay, first one, stem height. Some dominant were tall, recessive were dwarf. Second one, flower color. Dominant were violet, recessive were white in color. Third one, flower position. Dominant were axial and recessive were terminal. Pod shape. Pod means which encloses this pea seed. Okay, outer covering is called as pod. Some were inflated, dominant, recessive were constricted. Pod color. So dominant was green in color, recessive was yellow in color. Seed shape. Dominant was round, recessive were wrinkled in shape. Next, seed color. Yellow dominant character, green recessive character. Okay. Now, one by one. This is what dot. Tall, dwarf. Very important, given in the NCERT textbook. Dominant were violet flower, white flower was recessive. Flower position, axial and terminal flower. Axial means side, terminal means at the tip. Pod shape, you can see this one. Inflated, constricted pod. Okay. Now, pod color. Anikesh, my voice is audible now clearly?
Yes, sir, it's audible, sir.
Okay. Next, pod color. Okay. Green, yellow. Seed shape, round, wrinkled. Seed color. Yellow. Artificial pollination was very easy for Mendel to conduct. Selected people. Now, student monohydrate here. Just a minute. This is uh, for monohybrid cross. Okay, in your NCERT text, screen, same explanation is given in your NCERT book. Okay, what is monohybrid cross or single gene inheritance? Okay, definition is cross between two pea plant with respect to one contrasting character is called as monohybrid cross. T, capital T into T small. Always writing this very important. Parent always you must have to write this one here also. Homozygous. Now, gamete so capital T parent gamete. Always you must have to circle this capital letters and this small letters. Okay. Now, F1 progeny, first filial generation also we can write. Always cross capital T, capital T. Circling the parents is also important. Gamete, capital small t, capital T, small t. So F2 generation. Okay, I'll use Punnett square. RC Punnett, okay, was the first person to discover this Punnett square, or we can call it as checkerboard also. Okay, and right here only, and right here little bit F2 generation. So this capital T I'll write here. This small t I can write here. This capital T I'll write here. This small t I'll write here. So capital, 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 small, capital, small, small, small. So the phenotypic ratio, PR I'll write. What is this phenotypic ratio? It is the ratio expressed. What is phenotype? Morphological appearance of the organism is called as phenotype. What is genotypic ratio? Or genotype? Genetic makeup of an organism is called as genotype. 1 mark question, this will come. Okay. Next, what is homozygous? If both the genes are identical, it is called as homozygous. If what is heterozygous? If both the genes are non-identical to each other, it is called as heterozygous, we call. Is it clear? Next, phenotypic ratio is 3:1. 3 stands for tall. You must have to write. And 1 stands for dwarf. Next, genotypic ratio. 1:2:1. Only some students, what they will write, they will write ratio, but they will not write the full form of the ratio. 1 stands for homozygous tall. Homozygous tall. 1 stands for homozygous tall. 2 stands for heterozygous tall. Heterozygous tall. And 1 stands for homozygous dwarf. Homozygous dwarf means 1 is this one, homozygous tall. 2 is this one, heterozygous tall, and 1 is homozygous dwarf. Is it clear? So monohybrid class or 1 gene inheritance. Now, students, they have asked in the board paper, what will be the okay, phenotype. I'll write short form P and G, genotype, ratio of a cross between, this question has come, okay, capital T, and crossed with capital T and small t. Can you tell me the answer? I'll write the answer. If you can raise hand and tell me the answer, you can tell me before that, solving this one. So in a simple way, shortcut method, I'm going for shortcut method. But in exam, you should not use shortcut method in theory. I'm directly giving the answer. So phenotypic ratio is all are tall. All are tall. Next, genotypic ratio is 1:1 or 2:2 also you can say. What does 1 stands for? Homozygous tall. And another 1 stands for heterozygous tall. So this is the answer. Is it clear? So directly I'm solving the answer. Okay. One last question. Let me see how many of them are clever in this question. This has also come for the Cbse board. 4-mark question. Okay. The question is write the phenotype and genotype ratio of F1 generation where parents are heterozygous tall. If you are really clever, answer I can write here 3:1. Genotype is 1:2:1. Sorry. 1:2:1. 1:2:1. Okay. So what does 3 stands for? Tall. 1 stands for dwarf. What does 1 stands for? Homozygous tall. 2 stands for heterozygous tall. 1 stands for homozygous dwarf. So this is the answer. Okay. Now let us go to the next one. We'll go back to the slide. Okay. Again, selection of Mendel. So this is the definition: monohybrid cross or single gene inheritance. First filial generation, F1, means obtaining the progeny from the parent is called as first filial. See, F1 generation, this is first filial. This is second filial generation. Same in explanatory chart. I just now explained in the table format. In the explanatory chart they are given. Okay. Here, phenotypic ratio given in a 1/4 means 1 is dwarf and 3 out of 4 is tall. Tall. This is phenotypic method of writing. Okay. This is very important. One more question in your NCERT book. It is given. What are the unit of inheritance? Answer is gene. But Mendel didn't know gene word. We used to call gene as a factor. So we used to call it as factors. Because at that time, technology was not advanced. So we used to call transfer of factors from parents to the offspring. Like that he used to say. Nowadays, we call that one as genes. So genes are the unit of heredity. This one also have been a genetic heredity and variation. Heredity means transfer of parental character to the young ones. Factor means trait determining things. See here, factor means gene. Trait means particular expression we call, determining things. Like contrasting character traits we call. This is very important, student. Allele. In a simple word, alternative form of a gene, but exactly according to the genetics, we should not say. You write exam, what is allele means alternative form of a gene, but I'll explain that one properly because see, this is the chromosome obtained from the father. This is another chromosome obtained from the mother. So this is first pair of chromosome. There are 23 pairs of chromosome in our cell, each cell. This is the first pair. How we get this from father? Through sperm. How we get this one? From mother through egg. So when this sperm and egg fertilize, we are obtained. So imagine this is first pair. So can I call this one as homologous chromosome? Together if they are there means homologous chromosome I will call. I am explaining for allele. Now in this chromosome inside there is a chromatin network. In the sense DNA, coiled form. Imagine this part of the DNA in father, the same arm and same region in mother is controlling the skin color. My skin color is brown. That part of the DNA in father and mother is controlling this one. So these are alleles. So how to explain? Alleles are the genes located on a loci. Loci means region on a homologous chromosome which control one contrasting character, that is skin color. This is how the definition of allele. But if you are feeling difficult, alternative form of a gene, you write that one. Alternative form of a gene. Now, let me come back to the slide. So these are all our questions. Phenotype, morphological character appearance or physical appearance of the character is called as phenotype. Genotype means genetic makeup of an organism or genetic constituent of a character. Next, homozygous, explained. Two identical genes or alleles are called as homozygous. Capital T, small t. In one of the questions, Cbse board question, they have asked, human male sex chromosome is homozygous or heterozygous? Answer is heterozygous. And repeat, sex chromosomes are there in humans. There are two types of sex chromosome, XX female, XY male. So XX means homozygous, XY means heterozygous. So male sex chromosome are heterozygous, female sex chromosomes are homozygous. Okay, now this is important, but as it is not given, only test cross they have given. Back cross means crossing of F1 progeny with the parents is called back cross. Let it be dominant or recessive, we are not but. So we should not take this back cross. Test cross is important for you. Crossing of F1 progeny with recessive parent is called as test cross. Is it clear? Now, what is dominant character? Dominant gene also they can ask. One more question. Are those genes which completely express over recessive gene? Dominant genes are those genes which completely express over recessive gene. Okay. Monohybrid cross and dihybrid cross. Mono I said already, dihybrid cross means cross between two pea plant with respect to two contrasting character is called as dihybrid cross. Now recessive genes or character. Recessive genes are those genes which are suppressed by the dominant genes. Like that also you can write. Okay. Locus, position of the gene on the chromosome. Allele, alternative form of a gene you can say. Okay. Dominant allele are always in capital letter. Recessive alleles are in small letters. Capital T means tall, dominant allele. Small t means recessive, dwarf allele. Okay. Here, homozygous, heterozygous, homozygous. Okay. Leave this part. Okay. So this is another explanation. Homozygous and heterozygous. So again, repeated stem character. Now, genes, Mendel used to represent it as a factors. This also, a locus means position, already explained. Homologous chromosomes means explained. Both the chromosomes are identical to each other. Okay. Again, this one I have explained. So it's a repetition. Heterozygous means capital T, small. Homozygous means capital T or small t, small t. Recessive one which is suppressed by the dominant gene are called as recessive gene. Again, dominant and recessive characters. 7 contrasting character observed by the Gregor Johann Mendel. Okay. Now, So, yeah. Explanatory form of, okay. So this heterozygous, in some cases it is different. So let us not take that one. Punnett square. This is very important. 1 mark question. Reginald, this Punnett square. What is this Punnett square? Is also called as checkerboard. Tabular column representation of the progeny is called as Punnett square. We call that one. 1 mark question. This is 1 mark question. So this is what Punnett square explanation. Representation of progeny in a tabular form. Okay. So Mendel probability, as he was good in mathematics, based on the probability range only he could discover. Okay. So that method only he used in pea plant method. Okay. So how he calculated this phenotype and genotype ratio? Based on the probability method. So only he calculated. Here. Same. Here. We used to say probability 1 out of 4 were captured in monohybrid cross. 1:2:1 I said. So probability 1 out of 4 were homozygous small. This is important. In textbook they have given NCRT. Okay. 1 out of 2 were heterozygous condition. Or 2 out of 4 we can say. Instead of this one, if you are getting confused, 2 out of 4 are heterozygous. And 1 out of 4 is homozygous form. So 1:2:1 ratio. Okay. Okay. So same, that probability ratio explained in your textbook like this only. 1 homozygous star, 2 heterozygous star, 1 homozygous dwarf. Mathematic method he explained. This equation is given actually in your textbook. Same. Formula method, but they have given explain only 1:2:1 ratio. That's all. Okay. Now let us come to the next concept, test cross. 2 marks question. Crossing of F1 progeny with recessive parent is called as test cross. Why did he use test cross? They will ask for 2 marks. Significance of test cross. Sorry. What is test cross? Write its significance. The first is crossing of F1 progeny with recessive parent is called test cross. Significance to determine between homozygous and heterozygous condition. And write it and show it to you. Okay. Here. Okay. See. Capital T, small t into small t, small t. So F1 progeny is this one. Recessive parent is this one. This is a test cross. Okay. Now, what you will get directly, I'll write the progeny. Capital T, small t, small t. So capital small, small, small. So you will get 1:1 ratio. See, in a simple way, if I keep, see, this is tall plant, correct? This is also tall plant. Correct? So Mendel was feeling difficult to identify this tall plant, homozygous and heterozygous, because he didn't know what is that DNA or factor. He knew there is a factor, but he couldn't see the factors. Capital T, capital T, he couldn't see. DNA, he couldn't see. So he used to get confused. Who is homozygous? Who is heterozygous? To overcome that problem, he did this test cross. That is the significance. If you cross capital T with small t, you will get all are tall. But if you cross heterozygous with homozygous dwarf, 1:1 ratio. So 1:1, one tall and one dwarf. So what is the significance of test cross? To determine heterozygous and homozygous. This is the answer. So the same thing they have given here. Back cross, it is not given in your NCERT book. I am not going to. Okay. This is important. What are the three laws discovered by Mendel? Law of dominance, law of sum, law of independent assortment. I've given only two. And third one is law of independent assortment. Now coming to the law of dominance and segregation. Okay. Wait, I will explain in an easy way. Law of dominance. 2 marks question. Simply small t. Okay. Now you write gamete. In gamete, what you are writing? Capital T, as both the gamete are same. Capital T, small t. Okay. F1 progeny you write. First filial generation. So what you will get? Capital T, small t. This is tall. You know that. Now this is called as law of dominance. According to this law, the dominant genes completely express over the recessive gene. This is what the definition is according to law of dominance. Is it clear? And this chart, if you write, you will get the 2 marks. Okay. Next step. So, what are the characters of this dominant? This is given in your NCERT textbook. Are controlled by discrete unit called factors. Always factors occurs in pair. In a dissimilar pair of factor means capital T, small t, pertaining to the character, one member is dominant and another one is recessive. Capital T, small t. So these 3 points you should, what are the characters of law of dominance means 3 points you must have to write. It's given in your NCERT book. So monohybrid cross they have taken 3:1, 3 tall, 1 dwarf. Explain in a simple way, okay? So yeah. In law of segregation, what you must have to write? Capital T into small t, small t. Write this as parents. Now, write this as gametes. Separately, you should put a circle to this one. Right? According to law of segregation, the gametes get separated. Can you see? I have separated. I have separated, which are identical to the parent. I have written capital T here, capital T, capital, small, 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 small. So this is what you must have. The gametes get separated from the parents and they are identical to the parent. Did I write like this small t here and did I write here capital T? No, this is wrong. This is not a law of segregation. This is correct. They get separated and they are identical to the parent. This is called as law of segregation. Is it clear? Okay. Next. So this is what they have given segregation. Here, homozygous parent produce all gametes are similar. Capital T capital T means all are similar gametes. If it is a heterozygous parent means they are dissimilar. Capital T, small t. So this is what segregation. Now coming to incomplete dominance. After that, dihybrid cross I will go. Okay. Mirabilis jalapa, snapdragon. In this plant only they have observed incomplete dominance. Dog flower also you can write. Snapdragon also you can write. So what is this one? Also called a scientific name, Anthurium species they have given. Majuus I have given in textbook, Anthurium species they have given. See, this is our red color flower and white color flower. When you cross these parents, you will get pink color flower, hybrid. Sorry, heterozygous, I should say. This is homozygous, capital R, homozygous, small r, small r. When you cross this one, F1 progeny, capital R, small r will be pink in color. Incomplete dominance. What is this explaining? The dominant gene is getting blended with the recessive gene. Means here, dominant gene is not completely expressing over recessive gene. Okay, so that is why it is called as incomplete dominance. So here it is the extension, F1 to F2 generation. So if you cross, okay, I'll write it and show it. This is also is there for Cbse, one of the question paper I saw. The question was, what will the progeny of heterozygous in snapdragon? The parents are heterozygous here, in the sense pink in color. So this is the parent, this is the gamete, capital R, small r, capital R, small r. Right? Now, F1 generation, if you take out, checkerboard we will write capital R, small r, capital R, small r, capital, small, small. This is red in color. These two are pink in color. So one is red, two is pink, and one is white. Let it be phenotypic ratio or genotypic ratio. Both will be 1:2:1. Is it clear? Now coming to the codominance. Okay, Before going to codominance, dihybrid cross I will explain. Okay, I'll write it in a white board only. Students, cross between two pea plant with respect to two contrasting character. Okay. Now, here, according to your textbook, capital R, capital R, capital Y into small r, small r, small y. What does R stands for? Round. Capital R stands for round. Seed. Small r stands for wrinkle seed. Y stands for yellow color seed. Small y stands for green color seed. This is very important. So this is called as parent. Now this is parent. Now if you remove the gametes, capital R, capital Y, I'll write only one gamete, two gametes I should write. So one gamete. Now if I go to the F1 generation, what I will get? Capital R, capital Y, small y. Heterozygous round and heterozygous yellow seed. This is what F1 progeny says. Okay. Now, this is the F1 progeny. Now, if I do selfing of the F1 progeny, another heterozygous round and yellow, if I take out, so what are the gametes obtained here? The gametes obtained are capital R, capital Y, this is the first gamete. Capital R, small y, this is the second gamete. Small r, capital Y, third gamete. Small r, small y, fourth gamete. So these are the gametes obtained. Here also same thing I will write. Okay, what I have written here, the same thing I will write here. Now let us go to the F2 generation. Okay, I'll rub little bit part because I need a more space here, this side. Okay. Now, let us go to the F2 generation. I'll write it here. 4 4s are 16 box I must have to write here. That's why I'm writing F2 generation here. Okay, little bit mistake I did. Let me make a big box. Two, so 4. 1, 2, 3, 4. Now I'll take this gamete, capital R, capital Y. I'll take this gamete here, capital R, small y. This gamete, small r, small y. This one, sorry, I made a little bit mistake. Small r, capital Y, this should be. And last one, small r, small y. The same thing, capital R, capital Y, capital R, small y, small r, capital Y, small r, small r. Now here, capital R, capital R, capital Y, capital Y, right? Now here, capital R, you must have to write this chart for dihybrid cross as well as law of independent assortment. There also you should write this chart. smaller, capital Y, small y. Capital R, small r, capital Y, small y. Capital R, small r, capital Y, small y. Capital R, small r, capital Y, small y. Capital, capital, capital Y, small y. Capital, capital, small, small. Capital, small, small, small. Capital, small, capital Y, capital Y. Never my handwriting was like this before. Capital R. Writing in a tab is also totally different method. Such a bad handwriting this time. Okay. Okay. Now, phenotypic ratio. If you take phenotypic ratio, 9 is to this formula, you know that easy. 9 is to 3 is to 3 is to 1. Okay. 9 is to 3 is to 3 is to 1. What does 9 stands for? Round yellow. You should write this chart properly. See, number 1. 1, 2, 3, 4, 5, 6, 7, 8, 9 joining. So round yellow means 9. Okay. Next 3 means round green. This is round green 1. Okay, I'll use another color. I'll write here round green. This is 1. This is 2, round green. This is already counted. This is another round green. So I'll join this one. I have not counted this one. So 3 are round green. Next, let us go to the next color. Can you see this 3? Wrinkled yellow. This is 1. Wrinkled yellow. 2. Wrinkled yellow. So I'll join this one. Next. Number 1. Wrinkled green. This is wrinkled green. Can you see 4 triangles? Like this only you must have to write the checkerboard. You cannot change the gametes. If you interchange the gametes, you will never get the triangle. Is it clear? So this is how dihybrid cross is written. Now I'll teach law of independent assortment. Simple way. When a recessive gene get a chance to express, it completely express. This is what. Okay. So what independent assortment is saying means only this point is important. But you must have to write all the chart. But explanation stands in this wrinkled green. So here, this is the recessive of all, small r, small, all the 4 genes are recessive. But if you see the parents, heterozygous round and yellow, both are heterozygous round and yellow. So if you see 9 ratio, it is 100%. Same like parents. Correct? So 9 ratio stands for round and yellow. Correct. All are here 100% expression. If you see 3, round and green. So 3 is nothing but 50% of the parent is expressing. Only round is expressing. Green is recessive, it is not expressed. Another 3 you take here. What you are saying? Wrinkled yellow. Wrinkled yellow 50%. Only yellow is half of the parent. Wrinkled is not there here. Correct? Only yellow is there. 50%. Now 1 stands for 0%. They are not at all matching with the parent. Not matching like that. They are not expressed in the parent. Round and yellow is the parent. Heterozygous round and heterozygous. That is what here it is saying. How did this come? Recessive, when the parents are heterozygous round and yellow. It has come because of law of independent assortment. When a recessive gene get a chance to express, they express. So no dominant gene is expressing here. Is it clear? Okay. Now, codominance. 2 marks question. Human blood group AB. Don't get confused. Multiple allele is ABO blood group. Codominance means both the dominant genes expressing together. A blood group is dominant. B blood group is dominant. O blood group is recessive. If both A and B express together, it is called codominant. What is the definition? Both the dominant genes express together, it is called as codominant. In this line you can see here. AB, this is codominant. This is codominant. This dotted lentils and dotted lentils is another example. So, typical character as pleiotropic. One more question, pleiotropic. So one gene expressing multiple characters. So this one they have given in the NCERT book. Starch in pea plant. For example, starch synthesizing pea plant is controlled by one gene. It has two alleles, capital B, small b. Capital B is dominant, small b is recessive. Here, if capital B is there means large starch grain will be there. Small b means very less starch grain. Okay. Now there is one more, capital B, small b, intermediate size. Capital B, more starch. Small b, less starch. Capital B, small b, intermediate starch. So this is what. The same thing effect in the seed also. More starch, larger the pea size. See, medium means medium size. Small means wrinkled size. Okay. Now, concept of dominance. Again, law of dominance. Properties of dominant genes, this is given in your book, NCERT book. Normally, they are normal enzyme producing. Okay. And non-functional enzyme, no enzyme at all. These are the three genes of observed. Now here it has come dihybrid cross or law of independent assortment. Two gene inheritance also we can call. 5 marks questions to this. Seed color and seed shape they are taken. Seed shape round and wrinkled. Seed shape, seed color, yellow is dominant, wrinkled is recessive. So gametes, capital R, capital Y. Law of segregation. Here small r, small y. Now F1 progeny, heterozygous round, heterozygous yellow. Okay. Next step. Just a minute. Now 9:3 phenotype ratio, 3:1. Genotype ratio, 1:2:1:2, 4:2:2, 1:2:1. Simple seeing this one only, but they are not given in the NCERT textbook. Some students can write this one in examination to impress the evaluators in the marks. If some students write phenotype, it is okay. If you write genotype in a correct way, they feel that, oh, this student is clear cut in the dihybrid cross or law of independent assortment. Now, this is the thing. When a recessive genes get a chance, they completely express over 9:3:3:1. This is what very important. Out of 16 chances, only one chance it will get. Recessive gene, small r, small y, small y. This is what here explanation is standing. Here, this chart, Punnett square, I wrote already, same repetition. Just a minute. Now, this is very, in the textbook, they have given it like this. Independent assortment, when two pair of traits are combined in a hybrid, segregation of one pair of character is independent of other pair of characters. Okay, other pair of characters. This is the definition given in the textbook, NCERT textbook. Now, these are the explanatory parts. Capital so in theory exam, you can skip this explanatory parts. Writing the chart is very important. And this one, chromosomal theory of inheritance. What is this saying? Boveri and Sutton were the two scientists who proposed this chromosomal theory of inheritance. Okay, what did they say? They said that chromosomes helps in transferring of parental character to the young ones. Mendel said the transfer of parental character to the young ones. But Boveri and Sutton, they said It is the chromosome which helps in transfer of parental character to the young ones. Yeah, sure, definitely. Okay. There is one word here. Three scientists, Hugo de Vries, Carol Korn, von Teschmark, they rediscovered Mendel theory. This is a totally different explanation. It is given in chromosomal theory of inheritance. Hugo de Vries, Carol Korn, okay, von Teschmark rediscovered Mendel's theory. And based on this theory, Boveri and Sutton explained chromosomal theory of inheritance. See. Boveri and Sutton, chromosome play the major role. In which stage means meiosis stage. That's why linkage and crossing over came into the picture. Okay. Linkage and recombinant. So this is what Boveri Sutton. See how they are exchanging the segment. This is what they say. Walter Sutton and Boveri noted that behavior of the chromosome are parallel to the behavior of the genes and used chromosomal movement to explain Mendel law. In the sense, when they get crossover in meiosis, that time exchange of the segment takes place, DNA segment. Okay. Now comparison between chromosome. Always chromosomes occurs in pair, father and mother, homologous I said. Okay. Gene also same. So they are also in pairs. Now they get segregated during gamete formation. This also gets segregated. Only one of the each pair is transmitted to the gamete. Here independent segregation means they get separated. One pair segregation means the genes will get separated while expressing. Okay. This is important. Chromosomal theory. See here. Chromosome submetacentric as well as arcocentric. Before they were in pair. Now they have got separated in germ cells. So Boveri and Sutton, chromosomal theory inheritance was the major foundation for the Mendel theory. Where Mendel couldn't explain about, we didn't know what is chromosome only at that time. Next, linkage and recombination. One more question. Linkage means overlapping of homologous chromosomes. Recombination means exchange of the DNA segment between the homologous chromosomes. Here, this was experimentally proved by Thomas Morgan Hunt in an insect called Drosophila melanogaster. Very important. Experimentally proved by Thomas Morgan Hunt. So, Thomas Morgan Hunt. Drosophila melanogaster. Here. Here. Why did he select Drosophila for his experiment? 3 marks question. Easy to grow in lab, Male and female were easily distinguished. Single mating produced large progeny. So these are the things which made him to select Drosophila for the experiment. P.H. Morgan. So male and female. Normally female will be bigger in size, male will be smaller in size. See here. They could be easily grown in the lab and short lifespan, only 2 weeks. This one I forgot to explain. Only 2 weeks. And single mating produced large number of eggs or progeny. And he could identify male and female easily. So that's why he selected this one. And this one also. Variations were clearly visible under microscope. It is not given. We have mentioned that. Okay. So these things. There is one chart. I'll show the chart. Okay. Wait, I'll come to the chart. The chart is given in your here. This is important. Alfred Stewart Went. It is there in your book. Okay. He prepared chromosomal map. They have not given what is chromosomal map. It is nothing but representing the nitrogen base in a DNA. So like India map means what it will say, different state you will come to know if you see India map. So Alfred Stewart, what he did, Stewart Went, he made a chromosomal map. He used to say these genes are present in this area. Okay, like that he used to represent. This chart is there. This is what I wanted to speak. See student, this chart explanation is not there in the book, but one important question is there. Here you can see the cross. Okay, I'll go back. Okay. Now here parental type is 98%. Recombinant type is 1.3%. Don't take this number. What is this saying? Tighter the linkage, lesser the recombinant. Means if the recombination, if the overlapping is tight, less exchange of the DNA segment is less. So tighter the linkage, lesser the recombination. There is one more chart. Can you see this one? Recombinant 37.2, parent is lose the linkage, okay, more the recombination. That is what they are saying. Is it clear? This point is very, very important. Now mutation. One more question can be asked for 2, 3 in detail if they ask. Coming to mutation, sudden change in the genetic material, alter phenotype and genotype of a ratio, phenotype and genotype of an organism is called as mutation. Coming to this mutation, it was explained, recognized by Hugo de Vries. Okay, see here. This is the definition. Same thing I explained. Okay. In primrose plant, types of mutation, point mutation, frame shift mutation. Okay. Mutation can be addition of nitrogen base or deletion of the nitrogen base. This is primrose. This is given in the textbook, primrose. Hugo de Vries studied mutation in primrose plant. Point mutation. Point mutation means addition or deletion of a single nitrogen base in a DNA is called as point mutation. Okay. Single base pair in a DNA is mutated. This is called as point mutation. Example, sickle cell anemia. Normal shape RBC will be disc shape. Sickle means like this. Okay. Frame shift mutation. Shifting of certain nitrogen base from one place to another place is called as frame shift mutation. The deletion or insertion of the base pair of a DNA causes frame shift mutation. So this is frame shift mutation. These are the side effects of frame shift mutation. Here. See, can you see here? Shifting ATG. Now G is shifting to other place. So AAG. So this shifting is called frame shift mutation. Mutagen. It is the agent which causes mutation. Simple, one more question. Physical mutagen is UV rays, X-rays. Okay. These are all physical mutation. Chemical mutation, tobacco smoking. This is the only given in your NCERT textbook. Significance, it causes variation. Mutation causes variation. Now let us go to some of the MCQ question. Okay. The sum of total of character which is transmitted from generation to generation constitute heredity. Genetics is the study of heredity and variation. Heredity and variation are due to sexual reproduction, because transfer of parental character to the young ones it is taking place. Mendel conducted his experiment on Paisam Sataivam, garden tree, option 4. How many pair of contrasting character was studied by Mendel? 7. Which of the following character was not studied by Mendel? Length of pod, option 4. One of the following is incorrect with respect to experimental plant selected by Mendel to perform hybridization. It is a dicot. Sorry, it shows male and it shows male and female flowers separately. That's a wrong, unisexual. It's a bisexual actually. Incorrect it is, correct. Which of the following pair indicates recessive character in pea plant? It is wrinkled is recessive and terminal flower is recessive. Axial is dominant. Round seed is the dominant. Which of the following character in pea plant are not contrasting character? Round and yellow seed, correct. Round and wrinkled seed it should be, correct. Advantages of using garden pea for Mendel experiment include all list below except, true breeding variety was scarce. The main reason for the discovery of law of heredity by Mendel is all of the above. Emasculation is achieved by removal of anther. Alleles are homologous chromosome. alternative form of a gene. Correct. Alternative form of a gene. Oh, just a minute. Let me drink water. If a plant heterozygous for tallness is selfed, the F2 generation have both tall and dwarf. This proves the principle of law of segregation. An organism with two identical alleles of gene is called as homozygous. In a monohybrid cross, F2 phenotype is 3:1. The genotype of F1 heterozygous tall plant is capital T small t, option 2. Mendel principle of segregation was based on separation of allele in the garden pea during gamete formation. When an individual is having both the allele of contrasting character, it is said to be heterozygous. Law of dominance is the result of back cross. Okay. Which of the following law was discovered first by Mendel? Law of dominance. The term gene, which was coined by Johansen, was referred by Mendel as factor is the answer. Mendel produced pure white flowered recessive pea plant with a dominant pure violet flowered plant. The first generation hybrid from the cross showed all violet flower. Simple. Capital T, small t. All are capital T, small t. No? So like that here also, violet crossed with white. All are violet should be in F1 generation. In F1 generation, monohybrid cross dominant character appear in F2 generation. Recessive character will appear in which portion? 1:2, 2:1. 1 out of 4 is the answer. In a monohybrid cross, F2 progeny show 3/4 proportion tall and 1/4 proportion dwarf. From the group of tall plants, heterozygous. That is homozygous. Okay. Monohybrid cross, that is 3 out of 4. From the group of tall plants, 1 out of 3. Homozygous. Punnett square is useful for understanding it is the genotype and phenotype of offspring. Number of types of gamete produced by the monohybrid, always 2. Now, incomplete dominance. Red flower is crossed with white flower. All the offsprings are pink. This indicates incomplete dominance. Which of the following is incomplete? Snapdragon. In Snapdragon, when a red flower is crossed with white flower, the resultant hybrid plant have pink flower. If F1 hybrid is crossed with white flower, I'll write and show this one. Okay. Right here. So what they are saying here, in a snapdragon, red color flower. Okay. So this red flower is crossed with white flower. The resultant hybrid is pink color flower. So pink color means capital R, small r, is crossed with small r, small r. So what you will get? Capital, small, small, capital, small, small, small. 1:1. One is pink, one is white. So pink flower, this is pink flower, white flower, this is white flower. 1:1 ratio. Next. Complete dominance is absent in Snapdragon. In F2 progeny of Snapdragon, resulted through monohybrid cross, the cause of 1:2:1 phenotypic ratio is incomplete dominance. In case of incomplete dominance, F2 generation has genotype ratio equal to phenotype ratio. Just a minute. So there are only few questions. I will wind it up. In case of incomplete dominance, F2 generation has equal to phenotypic ratio. When two alleles of a gene are present and both dominant, the situation is called as codominance. Capital A, capital O, homozygous. Correct. Codominance. Both the dominant genes. What would be the color of F1 progeny as a result of cross between homozygous red? Pink. Alleles that produce independent effect in a heterozygous condition are called as codominance, which is called as pleiotropic gene. One gene, multiple character. They control more than one character. Pleiotropic gene, multiple phenotypic character. Option one yellow round. Okay, so let us go to the option. Now let us come to a true breeding. So this is short answers given. Pea plant homozygous for inflated green pods crossed with another pea plant with yellow pod with the help of Punnett square F1 generation. Simple. I'll write it and show. So inflated and constricted. Homozygous inflated means capital F, and green pod, small capital G, small g. If you cross small f, small g, F1 progeny, what you will get? This is what they are asking. Simple answer, capital F, small g. Capital F, sorry, 1 minute. Small f, capital G, small g. This is the answer. All are inflated and green. This is the answer. All are inflated and green. This is the. Now this is this one, pedigree analysis. On the basis of inheritance pattern exhibited, what conclusion can be drawn from this pattern? See here, affected. Father is affected, mother is affected. Father is affected, mother is affected. We cannot say mother is affected. Okay, here carrier they have given. So this dotted line means carrier, carrier male. So definitely it is a X-linked recessive disorder. Seeing this one only we can say X-linked recessive disorder. This is the first question answer. Okay. If the female is homozygous for the affected trait in pedigree chart, then what is the percentage of son will be affected? And if the female is carrier, okay, or affected, 100% son will be affected. Given in the genotype of offspring 1, 2, 3, 4 in the third generation. Wait, I will use the marker here now. So this is third generation. What does 1 stands for? It can be normal male or it can be affected. Number 2, 100% female will be affected. We sometimes study they are carrier female, but if both the sex chromosomes they get affected, that's why they are represented as like this. This is 100% affected male. 100% affected. Fourth one, normal female. X6. So it can be normal male or carrier male. 1 affected female, 100% both the sex chromosome are affected. Third one, affected male, normal. Fourth one, normal female. Okay. So thank you, students. Exact time, Akhilesh.
Yeah, sir. Thank you so much, sir. So today the biology session ends here.
Yeah.
So thank you for giving the knowledge to our students. I hope it was beneficial for them too.
Only few students.
Sir, like in between there were students. Now they are like it's
Tired.
8 o'clock, so they leave and they'll have something and come. That's it.
Okay, Anikesh. Okay.
Yeah, sir. Thank you so much, sir.
Thank you. Thank you.
Yeah, sir.